Hi, everyone, and thanks for joining me today for episode 746 of People Behind the Science. I'm your host, Dr. Marie McNeely, and today I am joined by Dr. Eric Skaar to talk about life and science. I hope you enjoy getting a behind-the-scenes look into the life and experiences of another researcher in today's episode. And listeners, if you want to meet even more of today's scientists, you can check out our website at peoplebehindthescience.com or subscribe to our show on your favorite podcast platform. And today, Eric is going to share some of his insights and stories, so get ready to meet another one of our outstanding people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am thrilled to be speaking with our guest scientist, Dr. Eric Skaar. So, Eric, welcome to our show today. How are you?
Doing really well. Thank you. Thank you for having me.
Well, thank you so much for joining us. And Eric, we're excited to learn more about you and the wonderful work that you do in our interview. But first, let me take a moment to tell our listeners more about your background. So, listeners, Eric is director of the Institute for Infection, Immunology and Inflammation, the Ernest W. Goodpasture Chair in Pathology, and Vice Chair for Research, and a University Distinguished Professor in the Department of Pathology, Microbiology, and Immunology at Vanderbilt University. He earned his bachelor's degree in bacteriology from the University of Wisconsin-Madison, his Ph.D. in Immunology and Microbial Pathogenesis from Northwestern University, and his M.P.H. or Master's of Public Health in Epidemiology and Biostatistics from Northwestern University. Afterwards, he completed a postdoctoral fellowship in microbiology at the University of Chicago before joining the faculty there at Vanderbilt in 2005. Now, listeners, Eric has received numerous awards and honors for his research, including being named an American Asthma Foundation Scholar, receipt of Vanderbilt University's Stanley Cohen Award for Excellence in Research Bridging Disciplines, the Pfizer Aspire Young Investigator Award, the Vanderbilt Chancellor's Award for Research, and more. He's also won a variety of awards for exceptional mentorship and teaching, including the Vanderbilt Molecular Pathology and Immunology Graduate Program Teacher of the Year Award, the F. Peter Guengerich Ph.D. Award, the Vanderbilt University Medical Center Postdoc Mentor of the Year Award, and others. In addition, Eric is an elected fellow of the American Association for the Advancement of Science and the American Academy of Microbiology. And Eric, we're looking forward to learning more about your science in our discussion today, but also more about you. So what do you like to do when you're not doing science?
I have a family who I love to spend time with. I'm fortunate to have two daughters. They're both juniors, one in high school and one in college, and try to spend as much time with them as I can, especially when the college daughter is home, which she is right now because we're in the middle of holiday season. And then when I'm not with my family, I really enjoy playing golf. And I also try to mountain bike pretty regularly. Those are my favorite activities.
Very cool. And what are your favorite family activities?
We mostly just like to eat. My wife and I like to cook and sit around and eat meals with our family and just kind of catch up on what everybody's been up to. That's kind of our favorite activity. And I think part and parcel of that goes anytime we can get a vacation that involves food, we particularly enjoy those.
Well, that sounds wonderful. And it's great to hear that you have some quality time with the family and things to look forward to outside of science. But you are doing remarkable work inside the lab as well, Eric. And I'd love to talk about your scientific side. So how do you describe your research to someone who's not in your specific field or someone who might be outside science altogether?
I consider myself a microbiologist and more specifically a bacteriologist. So we focus on bacteria as a group of microbes. Primary research interest of the laboratory is the impact of nutrition on infectious diseases. So we are interested in how what we eat affects our susceptibility to infection. And we're also interested in how the bacteria that infect us try to get food when they're inside of our bodies. And so that's the primary research focus of the laboratory, is this intersection between food and infection as it pertains to bacteria.
Very interesting. So there's some personal interest overlap as well in terms of the food.
You're sensing a theme.
Well, I think doing this exciting research can be motivational and inspirational in and of itself. But I think for those difficult days, it can help to have a quote or a saying or a mantra going through your head to keep you going. So, Eric, do you have a favorite motivational quote or something that really motivates and inspires you?
My favorite quote, it's actually painted on a canvas that's 1 foot to my left right now, hanging up next to my desk. My daughter painted it. It's our family motto, and it is work hard, be nice. It's our favorite quote.
That's amazing. So then is that something that you try to implement in the lab and in life every day?
Absolutely. That is my guiding principle, is work hard and try to be nice. And I view that as a pretty Midwestern attitude, which is where I'm from. I consider myself a Chicagoan, and I did a lot of my education in the Midwest. And that's how I was brought up. It's just work hard and treat people the way you want to be treated.
I love that. And I think you probably serve as a role model for a lot of the people that you interact with in terms of the trainees and the people that you're influencing on their own scientific career paths. But when you look back at your path, Eric, are there particular people who really mentored you or inspired you or who you looked up to over the course of your career?
And of course, a number of people really at each step, I think, throughout my career, there was somebody that in one way or another held my hand or lifted me up or showed me the right direction to move in. Depending on how much information you want, we could walk through all those people. But even starting as early as high school, I was fortunate to have a high school biology teacher that I thought really shaped my interests. And then that continued as I went into college and grad school, et cetera. So I've been very fortunate to always have strong mentors in my life.
Absolutely. And I think these strong mentors can have a tremendous impact, particularly in these early days of your career. You alluded to the fact that already in high school, you had this sort of blossoming interest in science. If we were to rewind back even farther, Eric, where did this interest begin for you?
I don't have a strong memory before high school of wanting to be a scientist. It wasn't something I wanted to be when I was a child. I wanted to be a magician or a fireman or something like that. But I always was pretty curious, and I always liked the natural world. I like to be outside, I like to play outside, and my parents encouraged that. And then I like to solve puzzles a lot. I always have liked to solve puzzles, and that I think is the primary reason I like to do science. I really enjoy the fact that the things that we work on are relevant to health and have the potential of impacting human health. But the real reason that I'm interested in science is I just like puzzles, and I like to try to solve puzzles. And to me, that's what biology is all about. And so it wasn't until I was in high school, I went to a public school in suburban Chicago, and I was fortunate to have a high school biology class that was half anatomy and half microbiology, which is very unusual for a public high school to have a microbiology class. But I think it was driven by the interests of the high school teacher. His name was Leo Lay. And Mr. Lay got me really interested in microbiology in that class. And I sort of never looked back, really.
Now, did you have a sense when you were starting to develop this interest in microbiology of what research was or what a career in research would look like?
Absolutely not.
Okay.
And to be frank, neither did he. So I was very interested in infectious diseases. I do know that. I remember I would grab the microbiology textbook and I'd flip to the part on infections, and I'd look at all the gross pictures. And that really interested me. And so I assumed, because it was the only thing that even kind of made sense to me, that I would be a physician who studied infectious diseases. I didn't have any physicians in my life. I didn't have any scientists in my life. So there wasn't really role models for me to look up to. But based on what I had seen on TV, et cetera, that just seemed like if you were interested in infections, you became a doctor. And so that was sort of what I went to college with the expectation of trying to do, was to obtain an MD eventually and study infectious diseases.
Very interesting. So then can you tell us maybe what were some of these key moments along your pathway? We mentioned in our introduction, you went to the University of Wisconsin-Madison for college. What really helped you kind of get on this path for research?
When I started in college, it's sort of a long story, but it's kind of interesting. So I'll tell it to you since I think we have some time together. I predated the internet as far as its ability to be very accessible. So it wasn't like we had college catalogs online where we could look up degrees and majors. And I decided to go to the University of Wisconsin because of its proximity to Chicago and its reputation as an outstanding science school. And when I arrived, I was very excited to begin majoring in microbiology. And the way they did it before registration online was we would sit across from another senior student with a giant paper catalog. And the senior student helped us identify our major. And so I told this person, who happened to be a band major at the time, I remember, that I was at the University of Wisconsin to be a microbiology major. And so she flipped through the catalog to M, and she couldn't find microbiology. And she proceeded to tell me that there was no microbiology major at the University of Wisconsin.
Oh, no.
Which was devastating to me because this is what I had built my entire career plan on.
Right.
So I started out majoring in zoology. Mm-hmm. And if anyone is listening to this from the University of Wisconsin, they're probably scratching their head that this doesn't make any sense. Because the irony here is the University of Wisconsin-Madison is probably the best microbiology program in the country. And it's so good back then, they subdivided the microbiology major into bacteriology, virology, plant pathology, mycology. So all of the different microbes had their own major. And the band major who was advising me didn't know that. So I got a slow start is the punchline of that, and ended up in zoology for a semester before I realized that the University of Wisconsin-Madison had an outstanding bacteriology program. And that ended up being what I majored in. And I needed money. And so I looked for a job in the science departments, something that a university position where I could make some cash to help make money. And the job that I identified was in the media preparation lab for the microbiology lab. So this was the laboratory that pours all the Petri dishes and makes all the test tubes with liquid media in them for the giant teaching courses in microbiology at the University of Wisconsin. So for the second half of the first year and into the second year, I was responsible for pouring thousands of Petri dishes so that the undergrad students would have Petri dishes ready when they showed up for their class. And the person who ran that facility, her name is Lynn Miguel, and she was a spectacular mentor to me. And she advised me after a few months in her prep lab and her hearing what I wanted to do for a living that working in a media prep lab was not the place to be if you wanted to become a research scientist, and you should actually get into a research laboratory. So that set me off on a course to try to identify a research lab to do undergraduate research in. And I didn't have bad grades, but I also didn't have straight A's. So I had a bit of a hard time getting people to respond to me and let me into their labs. But fortunately, Tim Donahue, who was a microbiologist at the University of Wisconsin-Madison, he offered me a position in his lab, sort of on a trial basis. I think that was at the end of my sophomore year. And that's where my introduction to research began.
Oh, very cool. And then when did you decide to kind of make that shift? I know for a lot of students, they sort of have to decide between the Ph.D., the M.D., or the combined M.D. Ph.D., or in your case, you did a Ph.D. M.P.H. So how did you make that decision of what you wanted to do next for graduate school?
I think a lot of this has to do with being exposed to the career tracks, which is one of the reasons why it's so important to get into a research lab if you're a young person who is interested in exploring research or being a scientist as a career. Once you get into a research lab, then you start to interact with graduate students and postdocs and research technicians and the faculty themselves, all of whom have been thinking about this a lot longer than you have and have much more experience in the career possibilities that exist. And so for me, I was fortunate to be in Tim's lab, and he had a full lab of people, all of whom had different career goals. Some wanted to be faculty, some wanted to start their own biotechnology companies. The Donahue lab is focused, or at the time was focused on single carbon metabolism in bacteria. And that led into a very strong interest in Tim's current interest in bioenergy. And so the people in the lab had really interesting career goals that spanned, like I already mentioned, starting new companies focused around bioenergy or moving into government or academic positions. So anyways, just having that level of exposure was really helpful to me to kind of solidify what I wanted to do for a living. And I realized that there are careers in research that don't involve getting an MD, that you can be a scientist for a career. And they sort of set me off on the track to get a PhD as the next step in that. And this sort of coalesced with my appreciation for many of the qualities that I think make physicians outstanding physicians are not necessarily qualities that I shared. I mean, like I mentioned, I was not really driven into research by the desire to help. I don't mean to sound like I didn't want to help, but that's not what motivated me. I was motivated by the process and the puzzles, and I like working with my hands. And so it just seemed to make a lot more sense that working in a laboratory, trying to make discoveries would be something I would be more interested in than interacting with patients and trying to help them be well. So that dramatically shifted my focus from what I thought I was going to do, which was an MD, towards more of a PhD directed career.
Interesting. And then what made you decide to go for that Master's of Public Health or M.P.H?
That's an interesting story. So I was in graduate school at Northwestern University, and I had one of the worst thesis projects that you can imagine.
Oh, no.
My thesis advisor, if he were sitting here, he would agree with me. He actually introduced me to give a talk in October a couple months ago, and part of his introduction was commenting on this thesis project. So it was nobody's fault. It was a good idea at the time, but in retrospect, we were sort of chasing ghosts. So the thesis project, quickly, the idea was to study how the bacterium Neisseria gonorrhea undergoes antigenic variation, which is the process by which it can change its surface to evade the immune system. And there was reason to believe that there were some genes in Neisseria that were responsible for this antigenic variation. And my thesis project was to chase them down, make mutants in the genes, and see if they were involved. And long story short is they were not involved, and there were more of them than we thought when we started. So I had to make many mutations in many genes, all of which had no phenotypes and were not interesting. And the reason that's relevant to your question is because I really didn't have any data until I was about a third or fourth year graduate student. So I started to panic a little bit and realized that maybe this dream of being a research scientist was not going to work out. So I began looking into what at the time we referred to as alternative careers. Now, these are the more common career than academia. This was right about when the book The Hot Zone had come out, and there was a movie called Outbreak that came out. There's a lot of really exciting things happening around infectious disease epidemiology and molecular epidemiology, meaning the use of DNA-based techniques to study the epidemiology of infectious diseases. And Northwestern University happened to have a part-time master's in public health program for the MD students, where they could participate in night classes and earn an M.P.H at night after their MD classes or at the end of their day. And I petitioned the institution to let me enroll in that same program, with the goal being to acquire a more diverse training portfolio so that I would be competitive for some of these molecular epidemiology jobs. Thinking like the Centers for Disease Control, EIS officers, and the EID fellowships that exist at the CDC. So that's what started me down that path. And so for the second half of my PhD, I took night classes with the MD students to get an M.P.H along with the PhD. And I'm glad you asked that question, because it is one of the things I'm more proud of, is that ended up becoming a dual degree program at Northwestern, because my experience was successful and we navigated the bureaucracy necessary to allow a PhD student to take the M.P.H classes. And now it's one of many dual PhD, M.P.H programs that exist in the United States. And there's been dozens of students that have gone through that. So that's a big point of pride for me, that that program has some staying power, and it was based on my experiences when I was there.
Oh, well, Eric, you were a trailblazer. I like it. So then how did you get then from that point where you were sort of struggling to figure out your next steps after your graduate school experience to where you are today at Vanderbilt? What were those key moments that brought you here?
I started to get a little more resourceful in the second half of my PhD, because even if I didn't want to do bench science, or it wasn't looking like a possibility for me, I still needed papers to graduate, and my project just was horrible. So I began to really kind of mine through the project graveyard of the laboratory, if you'll call it that. So we've all of us who've been in labs know that there's always unfinished projects or strains that were made that weren't used because of maybe the student who made them graduated before they could explore what their strain they made was capable of. And I started to pick up a few of those and just scramble and put together projects as best I could. And so with the guidance of my graduate mentor, who I haven't mentioned was an outstanding mentor, and I do mean to be critical of the thesis project because it was not good, but he was a spectacular mentor, and he helped me cobble together or sort of salvage my PhD in the second half. And I actually ended up with 4 or 5 papers in the end that made me realize that, wait a second, it wasn't me that was the reason I failed for 4 years. It was maybe the project. Mm-hmm. And it taught me this extraordinarily valuable lesson about the difference between a good project and a bad project, or a project that's likely to yield useful results and one that maybe is too high risk or unlikely to yield useful results. And that made me start to rethink whether or not a career in biomedical sciences was back on the table. And this was also happening at the same time that I was getting more engaged in the public health courses and beginning to realize that maybe that wasn't the career that I wanted, and that the outbreak movie with people jumping out of helicopters into the jungle to chase down Ebola is not really what all of those people do every day.
Unfortunately, right?
And when I started to get a better sense for how I might be spending my time if I was a molecular epidemiologist, it was less attractive to me than being a bench scientist studying basic science. And so I kind of repivoted and said, okay, I think I want to get back into this possibility of biomedical science career, and I'm going to do a traditional postdoc and see what happens. And so that led to a postdoc at the University of Chicago with the late Olaf Schneewind, who at the time was a rising star. An interesting part of that story is that when I applied to Olaf's lab, he was a professor at UCLA. And he told me during the interview that he was moving his lab to the University of Chicago. And he was one of 5 labs that I applied to, none of which were anywhere near Chicago. And so when I came home and told my wife at the time that one of the labs I had interviewed in was moving to Chicago, she was like, well, that's where we're going. And I agreed. So we stayed in Chicago and I worked in Olaf's lab for my postdoc. And that was a pretty significant scientific career change. I moved to more biochemistry and chemistry from molecular biology, and I focused on Staph aureus instead of Neisseria gonorrhea. And Olaf ran a very different lab than Hank's. Olaf ran a much larger lab, a lot more postdocs. There was a lot more energy around people interested in trying to launch independent careers. So I like to say that I learned how to do science from Hank, and I learned how to run a lab from Olaf, because my lab now looks a lot like Olaf's lab. And the scientific experience was completely different. So instead of having a project that I struggled with 4 years, I started out on a great project that Olaf really designed at the time, but it was my job to implement. And that project went extraordinarily well and extraordinarily quickly. And so I had a pretty short postdoc that was very productive, and that opened up all sorts of new opportunities. So that changed my mind and made me think that maybe I should try this academic career and try to run a lab. And that led to my faculty search.
Phenomenal. And I know now you are at Vanderbilt. Your lab is doing some really cool stuff. So I would love to talk about your science and what you've got going on lately. So do you have a favorite project that you are working on at the moment, Eric, that you want to tell us about in a little bit more detail?
It's hard to have a favorite project, like favorite kid.
Right.
We are involved in what I would consider to be the most expansive experiment we've ever done that I think is exciting and worth talking about. And that is, we are trying to identify the genes in phagocytic cells, particularly macrophages, that are involved in damaging the cell wall of bacteria. And the way that we are going about that is there's a very talented postdoc in my lab named Valeria Reyes-Ruiz, and she has created strains of Staph aureus that glow when their cell walls are damaged. And we're using a CRISPR library in macrophages, so a library of mutant macrophages, to identify mutant macrophages that can no longer damage the cell wall of Staph aureus and make them glow. And the idea here would be that we could hopefully find new genes in macrophages that represent new antimicrobial effectors or new cellular processes that are required for this phagocytosis, cell wall damage, and antibacterial killing that macrophages are capable of. And the thing that makes it so neat is it's a microscopy-based screen. So each macrophage mutant spanning the entire mouse genome is in a different well of a plate. So we have hundreds and thousands of wells representing all of these mutants, and then we robotically move a microscope to each well and take a picture to measure if the cell walls are being damaged by these macrophage mutants. And since we're taking pictures, when we get signals that tell us the macrophages are not damaging the wall, we can then go look at the actual cells and say, is it because these particular mutant macrophages don't engulf bacteria, or they don't form a phagolysosome, or they don't kill the bacteria, or whatever number of processes the macrophages are involved in? So without pictures, it's sort of hard to describe, but that's the experiment that's happening right now that we're very excited about, because it was just an extraordinary lift to get us where we are today, but it's working, and we should be done with the entire genome in about 2 more months.
Well, I feel like I'm going to sound like a grant reviewer here, but that sounds like a tremendously ambitious project.
Yeah, it's not the kind of thing that you would ask for money before you did it, probably. And also, it's one of those projects where if you knew how hard it was going to be when you started, you probably wouldn't have started.
I feel you on that.
But fortunately, Valeria is extremely perseverant, and we had enough success along the way to convince us that it could work if we just kept pushing, and we got there. So yeah, you're right. It's very ambitious.
And it sounds like you mentioned it's going to be wrapping up in the next couple of months here. So it sounds like a very exciting time. And I know these exciting projects and sort of heavy lifts in science have a lot of challenges and struggles and failures involved in them. It's just part of the process, I think. But most people don't get to hear about these difficult times in science. They just see the finished product at the end. So we love sharing these stories from all of our guests. So Eric, do you have a favorite failure story or just something you're struggling with right now that you can tell us a little bit more about and kind of share some of these struggles that people don't get to see?
Favorite failure story? There's so many. From a favorite standpoint, it's kind of hard because they all sting a little bit. And we've certainly been wrong a lot. That is its own kind of failure. You propose an idea or a hypothesis or a model, and those things end up being wrong. And I just think it's important as scientists to realize when the idea is probably not right and to back away from it. The expression we use in the lab is called punting. So when to punt a project. But when I think of the project, that I guess failure would be the most appropriate word that sort of haunts me the most. It's less of a failure like something didn't work, and it's more the inability of us to execute something. An example of that is I had a remarkably talented graduate student in my lab many years ago named Indriyati Hood. Her name is now Indriyati Hood Pashani, and she is a faculty member at the University of North Carolina, Chapel Hill. And when she was in my lab, she was working on Acinetobacter baumannii, which is a bacteria that causes pneumonia, and she was studying it in mice. And because getting an MD and a Ph.D. at the same time was not enough for Indri, she decided to also do an Ironman. And during the course of her training for the Ironman, the only way that she could perform her animal experiments were she had to do them at unusual times of the day. And when I say unusual times of the day, I don't mean like 6 a.m. or 6 p.m. I mean like 2 a.m. or 3 a.m.
Oh, my goodness.
And one day she came into me and she showed me an animal experiment where the mice had been infected with Acinetobacter baumannii, and she was showing the number of bacteria in the lungs of these animals. And the difference between the two columns was maybe 2 or 3 logs of bacteria. So one group of mice had 10 to the 6th bacteria in their lungs, and the other group of mice had 10 to the 9th bacteria in their lungs. And she said, what do you think the difference is between these animals? And the only thing I come up with was maybe one of them is neutropenic. Did you make a mouse without neutrophils? Because that phenotype is the biggest thing I've ever seen in an infected mouse. It usually means you broke the immune system somehow. So my guess is they're neutropenic mice. And the answer was not that they were neutropenic mice. It's that she had infected the mice, one group at 6 a.m. and one group at 6 p.m. or something like this. And that was the only difference. And that led to this hypothesis that maybe there's something about the circadian rhythm or the time of day when you infect these mice that is causing this susceptibility to infection to be so different. So we partnered with a lab here named Carl Johnson. He's a circadian rhythm biologist. And we began to investigate this the appropriate way, which meant that we would have to control the light-dark cycle for the animals and infect them at certain times of the day or night. We had to make sure they weren't exposed to light. So we had to procure night vision goggles so that Indriyati could infect the animals with the night vision goggles on. And it was just really neat and really exciting and really involved. And it was very clear that there was a circadian component to this phenotype. But the way to really prove that was to infect mice that don't have a circadian rhythm or have a defective circadian rhythm. And those are called clock knockout mice. And we couldn't get them at the time. Just they were too hard to get. They didn't breed well. The only places we could find them weren't either willing to give them to us or they didn't have them ready. And meanwhile, this extraordinarily ambitious graduate student had other things going on. And so she needed to get out of there. And so she ended up graduating before we could really lock this down and prove the phenotype was caused by the circadian rhythm. And to be frank, no one else has wanted to do infections every 3 hours, 4 days in a row. And so the project just died on the vine with this beautiful phenotype that we never were able to work up. And since then, if you were to PubMed circadian rhythm and infection, you would see a lot of Cell Science and Nature papers from people that have shown that circadian rhythm has a huge component in the immune response to infection. So failure might not be the best word, but that's what came to mind when you brought it up. We should have been right at that leading edge of those discoveries, but we just couldn't put all the pieces together. So that has still been a frustrating memory for the lab. But also, no one's worked up the circadian component to Acinetobacter lung infection. So that's still a viable project if we could get somebody who'd want to work those crazy hours.
Listeners, take note.
Right. I mean, I don't blame anybody for not wanting to do it. I wouldn't want to do it either, but it was a neat observation. So anyways, long story, but that's what comes to mind when I think of kind of the one that got away.
Well, this is a great story, Eric. And I think these missed opportunities are important to talk about in science as well, because the logistics often are a sticking point. You've got this great idea. You sort of have the approach planned out, but just that one piece, like you said, just getting that one mouse model that you needed was the sticking point that you really just couldn't execute on that plan. And that's something that isn't too uncommon in science. So definitely something that has to be considered when planning these different projects.
Yeah, absolutely. I think stick-to-itiveness is perseverance, maybe a better word, is one of the most important qualities in a scientist. And, this CRISPR screen in macrophages is something that exemplifies that. Valeria gave a lab meeting not that long ago, and she presented her screen. And as I mentioned earlier, we're over that hump where it's not working. And one of the other postdocs in the lab came up to me afterwards, just talking about how neat the lab meeting was and how cool Valeria's project was. And the comment he made is, very few people would have done this.
Right.
And I think it just kind of makes the point that a lot of science is just being willing to do things, try things, take risks, push in directions that maybe other people might not be thinking about doing or be willing to do. And that can often give you an advantage or an opportunity to make discoveries that otherwise wouldn't be made. So yeah, I absolutely agree. This is a big part of science.
Absolutely. We don't just want to talk about the difficulties, though there are many. We'd love to talk about the successes as well. So, Eric, do you have a success story, whether it was a big win or just a small but particularly meaningful victory in the lab that you'd like to tell us more about?
I think the most excited I think I've ever gotten in the lab was the first time I ran a PCR reaction and was able to run it on a gel in my own lab with chemicals and buffers that I ordered, and there was a band that I could see. That still stands out to me as the most exciting day in my lab was once I realized I could actually do something in my lab by myself, start to finish. Because as you know, when you start a lab in academia, you've given a room and a bank account, and that's it. And you have to make science out of that. And so to get to that first PCR reaction was just very rewarding. But that's not what you really asked. I think scientifically, the discovery that is the most rewarding that we're most excited about recently was we published a paper, I guess it was last year, 2022, where we described the identification of the first known zinc metallochaperone in nature. And this would be a protein that puts zinc into other proteins. And up to approximately 10% of the vertebrate proteome are zinc proteins. So up until this paper, it was unknown how 10% of the proteome that requires zinc obtains that zinc. And this paper where we discovered the first zinc metallochaperone was really rewarding for us. There were suspicions made by other scientists, not us, that this family of proteins might be the unidentified zinc metallochaperones. But when we unequivocally showed that these protein transfers metal to other proteins, I think that was probably the most exciting result that we've had in the lab, just because we knew the impact of that discovery to our field of metallobiology, that it was something the field had been looking for a long time. And we were very confident that the data were accurate. And that was just a really exciting combination of events for us. So I would say that's probably the most exciting time.
Well, that's wonderful. You've come a long way since that very first PCR, it sounds like.
Yeah. That was a big day, though.
Absolutely. So how do you celebrate the successes, whether it's these little wins that are particularly meaningful or these, like you said, big papers and projects that are successful?
We tend to celebrate people more than papers. One of the reasons is just the process of scientific publishing is so arduous and drawn out that by the time the paper is formally accepted, you kind of knew it was going to be accepted. You submitted your third revision 4 months ago, et cetera. And so it's just kind of hard to even know when to celebrate a paper because there's so many different milestones along the way where it's almost in, it's almost in, it's pretty much in, but not in yet. Now it's in.
Right.
And like, where do you celebrate that? And so instead, we celebrate people's individual successes. We celebrate everybody's, whenever they find out they get their next job, next position, whatever that is. We always celebrate that. We have certain traditions that we do as a lab to celebrate people's next step. We celebrate birthdays, but we don't generally celebrate papers. And maybe we should, but that's kind of how I've approached it, is more to focus on the people than the paper.
Absolutely. I think the people in science are absolutely remarkable. And I love this idea of celebrating the people and all these different milestones that they may have over the course of their time in your lab. And I think these traditions and these just fun and funny memories that you share with colleagues are often contrary to some of the stereotypes about what people think about scientists, right?
Yeah, absolutely.
There's a certain way science is portrayed in the media. So can you tell us a little bit more about these quirky traditions, or just share some of these fun human moments in science that show that we're not these sort of Autobots working in labs in white coats?
Certainly. Our lab tries to spend a lot of time together. We're fortunate to have a big group that floats somewhere around 20 people at most times. And that makes for a lot of personalities and I think a lot of fun. And some of our traditions include every birthday we go out for lunch and we celebrate the person's birthday. We also have a cake back at the lab, and whoever's birthday it was last is responsible for making the cake. So that leads to some very interesting culinary creations. Because as you can imagine, some scientists are much better bakers than others. Mm. And you can kind of get stuck with the wrong person being the birthday after yours. You're unfortunate. We always go out to pretty traditional happy hour type of thing for anybody leaving the lab. We work in downtown Nashville, and there's no shortage of places to have a happy hour in downtown Nashville. So that's a very common thing. Every year I host what we call the Northern Party. And what that is a party at my home where I prepare foods that are associated with the northern Midwestern United States so that I can introduce some of the people from the coasts and from the South to things like Chicago-style Italian beef or Chicago hot dogs or Wisconsin bratwurst.
Interesting.
So that is always a lot of fun. It's hard if you're vegetarian. There aren't a lot of northern Midwestern delicacies I associate with vegetarianism, but we try to accommodate as best we can. And then every holiday season, we just did this week, gift exchange, sort of a white elephant style gift exchange, where we exchange something that you can drink out of. That is the definition. It can be anything as long as it can be drank out of. So we just did that this week, which was fun. I got a glass shaped like a moose head. So that's pretty exciting. The other thing my lab does, which is neat, I tend to not get as involved in these, but once a month or so, they have what they call a taste test, where someone will pick a topic and they'll do a March Madness style taste off so that they can declare what's the best in a particular category. And those categories have ranged from cereal to foods you can get at Trader Joe's to seltzer water, carbonated water, etc. And that's always fun. And then they try to post the winnings on social media. And it just developed a little bit of a cult following of people that are interested in knowing what the lab has chosen as the best, dessert food that you can buy at Costco or something like this.
Well, I love all of these traditions. I think it does show just the character of the lab and the personalities of the people who are working in the lab, because I think these lab communities are fantastic. And I think having this great community of people can encourage you to really work really hard in the lab, but you also need to take breaks every now and then. So we do try to encourage our listeners to give their mind a break or to think about other things outside of their immediate science. So do you have a book that you can recommend that you've really enjoyed, whether it's related to science or not, that we should all add to our reading lists?
For science reading, I really liked The Eighth Day of Creation. That was a book that I really enjoyed. It's one of the few books I've ever read and marked up in my life. But as a scientist, one of the books that I mentioned this already, I'm aging myself. I predated the internet from the standpoint of being a trainee. And one of the big differences between people that are over 45 and under 45 is how we spend downtime in the lab. Because we did not have the ability to search the web and mess around and look up cool things, or there certainly wasn't things like TikTok and Instagram and all these distractions. So it was usually either a crossword puzzle from the New York Times, or you read a book when you were taking a break in between like incubations. And one of the books that stands out to me during that period of my life was called Eyewitness to History. And it's actually kind of a hard book to find. I bought it 3 times because I've lost it twice. And it is a book where it is 100 or so, hundreds of first-person accounts of sometimes mundane and sometimes extraordinarily important events that have happened throughout history. For instance, maybe just a diary entry from a person living on the plains in the 1800s, all the way to a first-person account of the pilots who flew the planes that dropped the atomic bombs in World War II. And I just find first-person accounts of events to be really interesting, be they, like I mentioned, extraordinary events or sort of daily events. And that book is very digestible because you can just read one first-person account in a 30-minute incubation or something like this. You can almost think of it like a series of short stories, except it's all nonfiction. So that would be a book that comes to mind that I would recommend to somebody who's looking to fill some gaps during a long day in the lab.
Well, excellent recommendation, Eric. We will add Eyewitness to History to our book list on our website for our listeners to find. And I love these books that have these little digestible chunks that you can sort of piecemeal attack. And we talked about some different aspects of your career, but one thing we haven't really touched on yet is travel. And I think this is a huge part of life as a scientist because you often do get opportunities to go to different conferences, to work with collaborators, and to present your work all over the world. So do you have a favorite place that you've traveled for science, Eric?
Yeah, this is one of the most underrated aspects of this job, right? So when I think about work and life and this idea of work and life balance, it's very important. And I think people absolutely need to take breaks. But I think one of the aspects of being a scientist that is great is there's so much life in your work as a scientist. And that's not just like passion around the research. What I mean is some of the most exciting places I've visited for work. Some of the best restaurants I've been to have been work dinners. Some of the most fun dinners I've had have been with colleagues who are from work, or I've met through work, who I now have known for 20, almost 30 years. So some of the places that come to mind, there's a Gordon Research Conference site in the Tuscany region of Italy called Il Ciocco that is a spectacular place to get to visit for work. And there's another Gordon Research site in Barcelona that has been one of the settings of one of my favorite trips of all time because one of my closest friends is another faculty member named Jim Cassatt. He's here at Vanderbilt. And my wife and myself and Jim and his wife went on a week-long trip through northern Spain before we got to the conference at Barcelona. And then Jim and I stayed at the conference at Barcelona. And that kind of opportunity, I mean, of course, we paid in personal money for the vacation portion of the trip, but the opportunity to sort of blend this trip together and get to see that part of the world and do it with a friend who I've met through work is just such a great example of how much a career in science can enhance your life and how there is this major life component to work as a scientist. And so long answer to your question, but probably the trip to Spain was my best work trip of all time.
Well, it sounds amazing. If our listeners are able to make it over to Barcelona or elsewhere in northern Spain, where do you think they should go, or what are some of the activities that they must put on their itinerary?
If you go to northern Spain, you have to go to San Sebastian. You have to eat pinchos, which are sort of like tapas, tiny little plates of food, and some of the best seafood in the neatest atmosphere right on the coast of Spain. So that would be a place I would add to a bucket list if you have any listeners that are interested in a trip to Spain.
Phenomenal. Well, listeners, we must add this to our list of places to go. And we know that you are addressing some major questions and problems in science in the work that you do, but oftentimes things stand in your way. We talked about some of the challenges you face, but often funding, staff, technology, feasibility, time, a willingness to do measurements or to do the experiment every 3 hours, as we alluded to, can hold you back. So if we took away all of the constraints and you were able to do anything, Eric, what is the one research question that you would want to answer most?
One of the things that I'm most fascinated by recently was a paper that came out. I think it was Grant Jensen's lab, who is a world-renowned cryo-electron microscopist. And I would have to dig up where this paper eventually got published. It was originally published on a preprint server bioarchive. And what Grant's lab does is cryotomography, where they take high-resolution images of cells, and the Jensen's lab was particularly good at taking high-resolution images of bacterial cells. So some of these beautiful structures that we've seen of type 3 secretion systems, type 4 secretion systems, et cetera, have come out of either Grant's lab or labs that do similar kinds of science. The paper that I'm mentioning was essentially a data dump of unusual images, structures that they saw during their hundreds of thousands of images that they've taken of bacteria. Shapes that don't make any sense, that are within bacteria, that do not align with anything that we currently know about bacterial structures or substructures. In other words, there's a lot of things inside of bacteria that are not part of our currently understood knowledge of the bacterial cell. And that has led me to get very interested in bacterial compartmentalization, or this idea that bacteria, like eukaryotic cells, have structures akin to or even identical to organelles, membrane-bound structures, that they use to potentially compartmentalize biochemical reactions. So the thing that I would be most interested in knowing are, what is the full library of bacterial compartmentalization or bacterial substructures that exist that allow bacteria to do diverse biochemical reactions? Because I think we've just begun to scrape the surface of this appreciation that bacteria are not just sacks of juice with proteins inside. And for me, it's a very basic science question, but I think I told you many times at this point in the interview that I'm interested in puzzles more than other things. And to me, that's one of the most fundamental puzzles, is what is going on in a bacterial cell? That would be my answer.
Well, I think that is absolutely remarkable. And I love the idea of just having this big archive of unsolved case files going out into the scientific community for people to start to figure out what is going on in these cells.
It's a cool paper. It's kind of like the CIA's UFO report, but inside the bacterial cell. Like, if you can hunt that down, if you're listening and interested, it's worth a read.
Oh, it sounds amazing. And Eric, we appreciate you sharing this with us today and sharing so much of your story and the insights that you've given us. But I'd love to end with some advice. So for listeners out there who are on their own journeys, what was the best piece of advice that someone gave you at some point, Eric, that really helped you?
The best piece of advice I have ever heard, and the piece of advice that I think has most profoundly impacted my career, is do not make decisions based on what would happen if you fail. Make decisions based on what would happen if you succeeded. That has really been a driving principle for me when I'm offered an opportunity where I'm trying to consider taking a risk. I say to myself, okay, let's not think about all the reasons this could go wrong and how bad it would be if it went wrong. Instead, let's think about what would happen if it worked, or if it went the way I wanted it to, or if I succeeded. And I found that if I take that attitude, it opens up a lot more doors for me to move into new and exciting directions and pushes me to achieve more than I think I would have otherwise. And so that is a piece of advice I try to pass on to people when people ask me the question that you just asked.
Well, I love this advice, this sort of leaning towards the optimism and not letting that fear of failure keep you from trying things.
Exactly.
Well, wonderful message to share with listeners. Is there any other last piece of advice or a last note of inspiration that you would like to end our conversation on today?
I think I would just say that a career in discovery in any industry is a remarkably rewarding career if you're an intellectually curious person. And I would encourage your listeners, particularly if there are some young people listening, to not be turned off or discouraged by what they may read on social media, because I have found that the voices on social media are particularly negative around careers in discovery, to the point of where they've actually sort of boxed out room for positive voices. And so those of us that love this job and love this career rarely even bother saying it out loud anymore, because the negative voices are so loud and often attacking. So I would just encourage someone who's interested in this career to not read too much into something they might read on social media. Instead, actually talk to the people who are engaged in careers in leadership and research in academia or industry or government, and ask them why they love their job so much and what the great things are about it, what the bad things are about it, and really try to get a true understanding of the career and not base career decisions off of some people that are just really loud on social media.
Well, wonderful message, Eric, and wonderful note to end our conversation on. So if our listeners do want to learn more about you, your amazing job and the research that you're doing, what is the best way for them to do so?
I think the best way would be to look at our website at the Vanderbilt University Medical Center. We keep a very active website where we have everyone that's part of the lab shown on there, and you can contact any of us, including me, at any time. And then contrary to what I did just say, I do try to maintain a social media presence. This is one of the reasons I'm aware that social media can be toxic, is because I'm on it. Mm-hmm. And so that we try to keep a positive social media presence that highlights the things that are happening in the lab on Twitter or X and on Instagram.
Phenomenal. Well, listeners, definitely get connected with Eric on social media. Check out that website to learn more about his work. And Eric, thank you so much for joining us today. It's been a true pleasure.
Thank you very much for having me. I appreciate it.
Well, it's been wonderful to chat with you. And listeners, great to have you here with us as well. We hope we'll see you again on our next episode of People Behind the Science.